Professor, tell me about the broad scope of the work that you're doing in your laboratory.
I'm a physicist and I work at the interface of physics and medicine, primarily with applications to ophthalmology. This includes development of optical and electronic devices for imaging, diagnostics, therapy, surgery, and prosthetics.
Tell us about some of the work that you presented here at ARVO 2016.
Yeah, we are presenting several projects. One group of talks and posters is related to laser therapy for what we call non-damaging retinal laser therapy, which we developed, and now we report not only animal results but also clinical, the first clinical results. It turns out that this therapy is broadly applicable to treatment of many macular disorders, including central serous choroidenopathy, diabetic macular edema, even macular telangiectasia, and we are starting clinical trials for prophylactic treatment of dry AMD to prevent progression of disease into wet or geographic atrophy.
What is the physiological basis of this approach?
So we found a window of thermal stress that exerts cellular response but doesn't destroy cells. And we developed a software that brings reliably in every patient laser settings from titration into this non-damaging treatment window. So cells respond to stress, and the thermal stress is a known rejuvenating factor. It has been demonstrated in many other organs, and we decided to apply it to the eye. However, I don't know yet all the details how actually from thermal stress and response with heat shock proteins that we measure, it translates into the benefits in so many diseases. For example, central serous choroidenopathy is a leakage from choroid into the subretinal space, and RP, the tight junctions in RP actually control it, and that may be one pathway. But it also helps with diabetic macular edema, where pathology is different. It's a leakage from intraretinal vasculature. And so we don't quite understand why it works in so many indications. For the upcoming prophylactic AMD trial, early AMD for patients with high risk of conversion, the logic is that we hope that thermal stimulation of RP cells will rejuvenate and enable them to recycle the drusen, the deposits that are accumulated with age, and thereby improve their function and delay or maybe even prevent progression. That's a hope, and we'll see if this will materialize.
Which of these areas is closest to translational testing?
This is all of these trials are starting this year. We are reporting preliminary case series, which was not controlled randomized trial yet, but we are actually starting multicentral clinical trials for central serous choroidenopathy and for prophylactic treatment of AMD with a center at Stanford and 5 other centers involved. Diabetic macular edema trial is also starting. It's also a prospective randomized clinical trial with a center in Manchester in UK. Paulo Stanga is leading that trial. It's still based on our technologies that we developed, and it is commercialized through Topcon, which manufactures Pascal laser. So all of them are very close to translation. They are actually in this transition from basic to research to clinical trials right now.
So what are your next milestones on this project?
The most important will be to see the results of clinical trials, and and this will take probably a couple years because the progression of disease, as AMD, is slow, and so we'll see probably enough statistical significance in 2 years. Diseases like central serous choroidenopathy are much faster, changes are much faster, and so we'll see the results within a year. We already have very encouraging results that we are reporting from 21 eyes this year, and so I have no doubt it will work. For AMD, we'll see. That's completely unknown. We are breaking new ground. And we'll also hopefully organize prospective randomized clinical trial for macular telangiectasia, which is probably also will be a long follow-up because it's a slow disease. But overall, it's very exciting times that suddenly we may have a preventative measure for dry AMD.
Tell us about the ways in which you're going to monitor outcomes.
AMD trial, the primary measure is the volume of drusen that we measure with OCT. The secondary is conversion rate into geographic atrophy of wet AMD, and the usual, visual acuity and just retinal thickness and all that. But these are the primary measures of outcome and ultimately success of this trial.
Tell us a little about some of the other projects that you're working on in your laboratory.
Another very interesting project that also is actually moving from basic research to translational phase this year is restoration of sight to the blind using photovoltaic retinal prosthesis. We demonstrated that this very thin arrays of photovoltaic cells, pixels, it's like solar panels that convert light into current, can be injected in subretinal space in rats, and they can restore visual acuity at the level of half of the normal in blind rats. And we demonstrated this now for life of animals. It's 1 year approximately. And this year, this technology is being transferred to clinical trial, which is scheduled to start in fall 2016, this year in Paris. We do it in collaboration with company Pixium Vision. It's a French company, and the trial will be in Paris. And I'm looking forward to these results. It was long coming. We started this development probably 10 years ago, and finally it comes to fruition with excellent results in animals so far, and we are looking forward to human trials.
What was the breakthrough that made it possible now?
Well, it was slow coming because fabrication is difficult. All the measurements take a long time, and just development from scratch, from idea 10 years ago to the product in this field of electroneural interfaces is a pretty high pace. Luckily, we found a partner who is willing to spend enough money that is required for clinical approvals and clinical trials, and this actually enabled translation of this technology. Otherwise, it would stay in a lab. It takes tens of millions of dollars to convert a prototype into fully functional and approvable by CE mark and maybe future in FDA product. That takes a lot of organizational and financial efforts, and luckily we found a partner that is doing it, and that's the company Pixium Vision.
Extraordinary, absolutely wonderful. Last question is how would you phrase this work for patients? What can you tell patients about the implications of what you're doing?
I would say that we are trying to address age-related macular degeneration and retinal degeneration in general in two ways. In cases where we can prevent it, like an AMD, we are working on prevention, and this is a laser therapy that hopefully is a prophylactic treatment that will rejuvenate RPE cells and allow them to recycle accumulated deposits, drusen, and thereby prevent progression into the loss of sight. For cases where the loss already occurred, photoreceptors are lost, we are developing a second strategy, which is a restoration of sight. And so this is a two-pronged approach, early intervention and late intervention.
Excellent. Professor, thank you so much. That
Absolutely.
was really excellent. I really appreciate it.
Thank you.